There we go. Because we will hopefully be having some people join. Yeah, I'll kind of like wander around as you move. The only problem is people get like a shot straight up my nostrils when I'm trying to figure out what's what. Yeah, cool. Yeah, so I'm sure we've wanted to go back to Ecuador. Like we haven't been there since 2003 for a whole variety of reasons. And I know that if I go back and I'm just immersed back into that language again, it will start coming back. You know, because I can read it fine. Speaking is different.
Okay, thank you. Thank you for inviting me.
Hello, Marv. You're equipped with your diet, Dr. Pepper, as usual? Yes. From to Ecuador? I don't know. We've not actually looked into it because until recently we haven't been in a position to actually leave. I think most flights go out of Miami. That's the main route to Ecuador.
No, it's pretty affordable. I'm always checking flights.
Mhm.
And if you're not going in high season, you can find flights for $500.
Well, there's no seasons in Ecuador. It's like it's always the same. Slightly rainier or slightly drier, but that's pretty much it.
We're in this moment in our life that we can't afford traveling, that we don't have the time to do it.
Right.
This is the first year sometime in the US that I'm actually taking a ton of holidays.
Wow.
And European styles.
Mm.
Several weeks. I tell my students, my phone is not working.
Well, good. No way. Uh-huh. Whereabouts are you going?
But to Bogota, from Bogota, we're going to go to the East Coast, me and my brother.
To Cartagena?
Not the southeast coast, but it's not as known.
Okay.
And then that's the first week. And really by the second week, we will move north to Cartagena and Santa Ana. And then look at
Santa Marta.
Santa Marta.
Yeah, there you go.
But it's not push waves. And I'm more like a hiker and parks and things like this. So, well, I'm still trying to figure it out. I'm going to be in a natural reserve in that area.
Cool. Yeah, it's a beautiful part of the world, that's for sure.
Yeah, so I'm very excited.
Mm-hmm.
And I was checking flights like a year ago, and I started noticing that you can find such cheap flights if you buy in advance.
Right.
There's no take advantage of that. My brother pays double, of course, from Spain to Colombia.
Right.
Because I thought this was closer than it's
You get-
North America.
If you get the chance in Bogota, the museum, I don't know what it's called actually. It's like the Gold Museum. Is really cool. It's like in the central bank vault. Like there's this whole museum of gold, like all like pre-conquest and post-conquest gold artifacts and like the myth of El Dorado and all that sort of stuff. Mm-hmm. Yeah, it's really cool.
Yes, the big causes of the
Yeah, if it's still there, I mean, that was like 20 years ago or more. Mm-hmm. Yeah. It's a big, cold, nasty place, in my opinion. Don't like it very much. Yeah, absolutely terrifying airport as well, just as a heads up. Hopefully you're not. Yeah. All right, we better get going. Davida, would you like to introduce our speaker? Oh, you're not going to read through all of that, are you?
No. So, folks, I'm really excited. Marv and I are co-hosting the speaker today. She's an epic woman in STEM, and we don't meet too many of them, so I think it's delighted me. She's tenured. She's an associate professor of biology at Texas State. Down the road, Texas State has all sorts of lovely aquatic programs. She completed her PhD in biology at the University of Barcelona and a postdoc in the Centre for Marine Science in Portugal and in Michigan State University. She's been around. Her research, I have to write this down because I won't be able to say it. I don't understand half the words. Her research interests are driven by her fascination for the pheromones and endocrine systems of fish. She studies how chemical signals drive reproductive output and has investigated the activation of the endocrine axis after a pheromone detection event. And that's why I study bacteria, because that's too much for me. She has expertise in electrophysiology, and she studied using the physiology of the elements of the hypothalamus, pituitary gland, by protein biochemistry, histology, and molecular methods. She's published dozens of papers and presented her work in the US and internationally, and she's also received numerous awards, including the Excellence in Scholarly Creative Activities in Texas State and the Teaching Award honor in 2020. And my favorite thing about her is that she's really looking forward to her sabbatical, which will bring her to the Kavli Institute in Norway, which is awesome, and the University of Varese in Italy. So welcome, Mara. We're delighted to have you.
Thank you so much. Hello, everyone, and welcome to my talk about the sense of the smell in vertebrates and how the environment is going to affect these senses. So let me just start my lab. So as David said, I'm at Texas State University, and my research is centered in the sense of the smell, using fish as a model. So I mainly study basic research and trying to understand how the sense of the smell work in vertebrates. But I also work in the ethical pollutants and fashion. So I try to apply my basic research into practical problems. I also try to understand new sense that we discovered like 2 years ago, in which fish seems to smell pathogens. And this electric signal is activating the immune system. And I'm also working in several production systems like aquaponics and aquaculture, and how chemical signals can help us to modulate these physiological functions that will increase the output of production of fish. So sense of smell. Let's speak about that. All vertebrates have a sense of smell. It's very common. And if you didn't know, fish have a fantastic sense of smell. All of us have this cavity in front of our face that is flowing air and water and take all these chemical signals to some neurons that are there just producing their dendrite and are detecting these changes in the chemical environment. But you might think that the sense of the smell is not very important, isn't it? And this is because for centuries, we thought that the sense of the smell was something very subjective, isn't it? Something that for me smells like roses, for some of you will smell like rotten fish. And it's related with memories, passion, and very, very subjective feelings, to the point that even philosophers in the 19th century was thinking about, this is the most incredible sense. Why should I smell disgusting things? And what is the meaning of the sense of the smell? And it was around the 1991 that we have a great discovery about the sense of the smell. We discovered that this sense have more genes than any other sense. So it's indeed crucial for humans. The sense of smell allows us to smell chemicals at very, very, very small concentration. And they're communicating so much information. So it was then, in 1991, I was in college as an undergrad, and I'm totally saying what is my age now, that it started an increase of research in the sense of the smell. And it's funny because I have this graph until 2019, and you can see the increase of papers. But you know what happened in 2020? COVID. And this is still a spike much more. This is because our sense of taste is linked to the sense of the smell. So when we eat food, and we're also breathing, we are kind of transporting this chemical molecules to our nose in what we call the retronasal flow. So everything we eat and we taste, it's actually not in our mouth. We have the sense of sweet, salt, and sour in the tongue. But this is smell of coffee, chocolate. And David, I have the coffee there. This is actually located in our mouth. When COVID happened, not only affected our respiratory system, it also affected our olfactory system. And here I have a nice figure from our colleague showing these neurons in the olfactory system that are in gray, and the sustentacular cells that were actually affected by specific receptors in the cells that are surrounded the neurons in the olfactory cell. That means that these cells die because the SARS-CoV-2. And when this happens, we lose our sense of smell. And some people, I think, you know that they lose their sense of smell for years. So we're starting to kind of appreciate the sense of smell as a human species, not only as fish biologists after COVID. But let's go back to fish, because this is what they do. You guys knew that fish actually have an amazing sense of smell? And they have a huge nose. So I brought something here to show you, because a student of mine, I'm going to show you the olfactory epithelia of fish. It's also in the nasal cavity. So if you remove the flap, the nasal flap, you can see this is every fish. You remove the epithelia that is just on top of the nose, and you can see the structure, lamellar structure of the box. And this is a representation of this. So every one of these lamellas are covered with olfactory receptors. And it's sometimes like a third of the surface of the fish. Fish actually have a very sensitive sense of the smell. It can smell like even more than a dog. And they use this scent, this highly sensitive scent, to coordinate a bunch of functions. So let me speak first about the molecular and cellular properties of the sense of the smell. Fish as humans also have different olfactory sensory neurons that are producing that leads to the outside media. But they have a different morphology than humans. Humans would have only 1 or 2 types of cells that are microvilli cells, but fish have 5 different types of neurons. They actually have a more diversified system of detection of molecules. And then these signals that are elicited by these neurons will go to the olfactory folder. If we do a section of this nose, of this rosetta, we'll see something like this, where these white spaces are the areas where the water is flowing, and you will see every one of these lines is actually a lamella. If I do a higher magnification of these lamellas, you will see here in blue, and these electric blue are actually the mucus cells that are producing these mucus that cover the lamella and kind of giving some kind of protection. And then every one of these little dots are the olfactory neurons that are producing their cilia. You can see here that this is hairs. This is the olfactory cilia that are involved in the detection of odors. And in the middle, I don't know if you can see very well, these lines that are actually the action of the neurons that are going to translate all this information to the brain. And the orange ones are actually red blood cells that are going to give oxygen to the nose. So why is fish important? Why the sense of smell is important in fish? And I wanted to put just very briefly an example of what happened during COVID times. During COVID times, everyone shut down, had to be at home. And I was like a Spanish witch in my cabin doing nothing, like poisoning apples, something like this. And a colleague of mine says we should do something with COVID. So we decided to see what are the effects of this spike in the nose. At that time, no one knew about that. And I was the only one able to go to the lab, so I come with a lot of having this research. And what you can see here is olfactory responses that I can measure with electrodes in the nose of the fish. You can see that if I have a response of 3 millivolts, that will be almost like 100% olfactory response. When I apply the spike, not the virus, only the spike, you can see an instant reduction, like in half minute, you lose 50% of bacterial sensitivity. This is how effective is the spike of this virus in our olfactory system. And this actually is maintained for a long period of time. Not only this, you can see here that when I try to measure these olfactory responses 3 hours, 1 day, and 3 days after exposure, the reduction of olfaction increases from 50%, 70% to 100% basically. I'm going to tell you something else. Day 3, I don't have here anything because in 3 days, several people recover their sense of smell. They have a faster turnover than mammalians. There's a recovery of bacterial sense. We also measured that this damage created by the spike was related with olfactory function. So when we put a piece in a 2-choice maze and ask the piece to decide to go swimming towards another that will be just water or another that is food, the fish decide to go this other end thing if they have a healthy nose. But after exposing to the spike, they lose their ability to find food. So this is a good way to convince you that actually fish are a good model to study olfaction. And not only this can be translated to human health, because they're basic elements of olfaction, neurons, molecular GPCRs that are involved in the detection of these chemicals are common. But let me tell you also why olfaction is important. Fish are important for several reasons, isn't it? They give us protein, they give us food, and they're also a part of our natural ecosystem, which we want, at least me, I want to preserve. So one of the most amazing things in terms of chemical communication of fish is pheromones. Did you guys hear about pheromones before? They're college students. Well, so sometimes it's funny. I'm from Spain. I wanted to tell about pheromones in a way of a telenovela. La feromona. And I just think about this phenomenon. This visualizing this as I start a blog. So goldfish are actually known because their mating system works with pheromones, this substance that sends this information about reproduction. So here is Maria. Maria, it's a female goldfish and she's ready. She's full of eggs and she has to release this soon. And this guy with a mustache, it's Rodolfo. Rodolfo is also ready. He wants to reproduce, this is the way of life. And sparkling water lasts like 1 minute. So how do fish know when it's the time to reproduce? You might think they see each other, isn't it? Did you guys see the color of the river here in San Antonio? Do you think anyone can see anything? The most part of our freshwater systems are turbid. No one sees anything. So Maria cannot see Rodolfo. What else can be a way of communication? Speaking. Goldfish are terrible speakers, so let's forget about this. Or touching, but fish actually try to avoid predators. They kind of hide. They live in very, very different, diverse ecosystems. And the sense of touch is kind of more related with detecting vibrations in the water. But of course, because they don't speak, they don't develop the sense of hearing. So that's it. This is a drama. They cannot find each other. The goldfish and all the fish species are dumb and they are going to disappear. But there's something else that we didn't take into account. Maria can be. And in this release of urine that she can control, she can put molecules that signal further stage in reproduction. So these molecules will go no matter the color of the water, no matter what are the obstacles, it will reach Rodolfo. And what will happen? So this is a pheromone, a typical example of pheromone communication. And pheromones are molecules that induce an avoidable physiological response. So they are powerful. Can you imagine if I have a human pheromone that induces reproduction? I will be rich. It doesn't exist. It's not on the internet. If they tell you something that is a pheromone, it's actually a pig pheromone. You will have a fantastic girlfriend in the pig farm, but it's not working for human. And it costs $80, so don't buy it. But this we actually use pheromones for the control of domestic and wild species. So the sense of smell in pigs is further the fact that the sensitive system of olfaction uses the sense of smell to coordinate reproduction, imprinting, migration. They can smell their own river. They can detect food. They can be alarmed by the smell of a predator or by some other beasts that are releasing alarm pheromones when they're attacked. And I like to think in the sense of the smell as a button connected to a computer that it will be our brain. So if we have a problem with our sense of the smell, you cannot press this button, you cannot activate the brain, and you cannot think about what is going on in your environment. And if you press the button, you cannot reproduce, you cannot eat, and in the long term, it's the end of the species. So when I came to Texas State, I was thinking, what can I study? What will be my line of research in this new lab at Texas State? I was thinking, we know very, very little about what's going on with our pollutants in our water. We're amazing species. As humans, we're incredibly creative and productive, producing junk that we put in the river and in an aquatic system. And one of them is nitrogenous compounds. Because we're growing as a human population, and this is a global problem. Because we need to grow a lot of crops once we are using a bunch of inorganic fertilizers that are full of ammonia. And this ammonia, when it goes to the water, is transformed in nitrate. So we have actually a regulation for the amount of nitrate we can have in the water. It's 10 milligrams per liter. And I can tell you, this is what the EPA wants. This is not what happened. You can see I put this map just to show you the amount of violations of this rule that is happening for, I think it's the last 10 years. I think they are triplicating. I have it here in terms of time. So we're going through 20% of violations to almost 40%. And Texas is one of the biggest offenders. So perfect. I found my area of research. So let's speak about how nitrogenous compounds can affect the sense of smell. And I'm going to speak about nitrite. And you can notice that nitrite, NO2 is not the same as nitrate, NO3. So I'm going to speak about it. So we have all this nitrogenous waste that comes from fertilization, from animal manure, human manure, so all the septic waste that go to the water. This nitrate is transformed by bacteria, generally this a group here, the nitrobacter, to nitrite. And also fish excrete their products from metabolism in the form of ammonia, not urea like humans, ammonia. So all this ammonium can be also transformed to nitrite. And actually, when you look at a fish farm, you can accumulate a huge amount of ammonia transformed into nitrite. So there are several ways to increase the amount of nitrite in the aquatic media. And probably if you measure nitrite in some of your tap water, you might find nitrite. You might be surprised. There's also another way to have an intake of nitrite when you don't want to. Our mouth and our stomach and our intestines can biotransform nitrate, NO3 to NO2. So if you have a huge ingestion of nitrates, you might have some health problems. So long time ago, like 20 years ago, when I did my master in the effects of nitrite in fish health, at that time it was a big problem in aquaculture, and it was one of the main problems for the development of intensive water systems. Fish feed and all this ammonia is transforming nitrite, and nitrite, very interesting, it oxidizes hemoglobin. And it oxidizes irreversibly. So it binds to this M group. And then hemoglobin cannot bind to oxygen anymore. In fish, normally this hemoglobin renders blood brown, and it triggers this what we call the brown disease. So you see this poor goldfish that we had the tail that is exposed to nitrogen for a while, and you can see, and if you don't see, this is brownish compared to this control fish that has this brilliant blue. But we also have cases of methemoglobinemia in humans, and in our case, we look like bluish. I don't know if you guys hear about blue baby syndrome. So it can happen that by some environmental accident, you are drinking nitrates and actually you have an episode of methemoglobinemia and your blood becomes blue. But this is not the only effect. There's also effects in your heart. So you do some heart damage, ionic imbalance. It's a chemical irritants role. Your mucus are producing mucus. And you have damage in your liver and mitochondria. But this is for huge quantities. I'm speaking about 10 millimolar, 20 millimolar. And what we find in the environment nowadays, due to agriculture, will be like 1 millimolar. So I was thinking, well, I know that 1 millimolar probably is not affecting fish physiology in terms of the blood is going to be okay, and digestion going to be okay. But what about the sense of the smell? You remember these neurons that are expanding their dendrites to the water, and it's going to be full of nitrates? What happens when you expose these neurons for a long term to nitrates? So that's how everything started. I exposed goldfish, that is one of my model species. It's a fantastic fish because I can have goldfish in any pet shop. And they grow well, we have the genome, they're friendly, they're cute, so they're fantastic model species. I exposed fish to control water, so 3 millimolar, a low concentration, 1 millimolar, they do fine, 1 millimolar, and 10 millimolar where they started having toxic episodes and symptoms. And then I analyzed how these nitrates accumulate in 3 different tissues: the nose, the brain, and the gill. And you can see that in the case of brain and gill, the first 2 treatments, there is no difference after 5 days. And of course, when you put a bunch of nitrate inside the water, it goes inside. But the nose was actually increasing the amount of nitrate. You can see this is different. It's a significant increase only in 1 millimolar. So it's accumulating in the nose only in 5 days. The reason it's accumulating is because there is an active transport that it was described or suggested like a couple of decades in the gills. We knew that the mechanism of toxicity of nitrite is by active transport nitrite through this exchanger here. This is an exchanger that is in the gill of fish. It's used to osmoregulate, to exchange carbonate and chloride in order to maintain their osmoregulatory process. And it says that nitrate can hijack this chloride transport and be transported inside the cell. So that's why at these high concentrations, nitrate is in the gill. And if it's in the gill, it's in everywhere. It's in the brain, it's in the nose, because it's in the blood, and it's going to all your tissue. But why is it in the nose? One is still not in the gill. It's still not in the blood system. So one of my hypotheses is that the nose is actually a transporter for nitrite. And I actually checked for this nitrate transporter. Well, it's a chloride transporter, and I can see that there is expression of this transporter in nose. So nitrate can concentrate in goldfish. Nitrate can be transported, and specifically in the nervous system. So what happened in the nose? And if you remember, this is a catapenosis. You can see every one of these is a olfactory lamella. This is the central part, and this white space is the interspace where the water flows. You can see again in electric blue, these are mucus cells that are generally in the outside of the lamella, and all this here is the sensitive border. So this is a control fit. I check in this 5 days exposure what happened at 0.3. And maybe you don't see very clear, but this is subtle effect. You can see these electric blue spots. increased compared to the control. So this is an inflammatory process that is starting in the nose. But I'm still not seeing anything happening in the fish. The fish was eating, it was happy, it was not any apparent effects in terms of the stress. So something is starting to happen in the nose. At 1 millimolar, that it's not, the fish are not dying, but they're stressed. You can see what is happening with the interspace of the olfactory lamella. They're almost collapsed, isn't it? And you can see also that the mucus, that it's this blue electric, is kind of covering these spaces. This is the equivalent of you when you have allergy. Yeah. You know, you still smell, but it's kind of uncomfortable, isn't it? This is what happens when you don't smell at all. This is a total collapse of the nose. So nitrate collapses the sense of the smell from very, very, very small concentrations. So it says, okay, I found something. I think what is happening now in our environments is dramatically affecting the sense of smell in fish. And remember, the sense of smell in fish is very, very important for growth and reproduction. So if I can demonstrate that these small concentrations of nitrates are affecting our populations in the long term, I will make a big advance, isn't it? But I wanted to do it from several points of view. I wanted to see what is happening in the immediate space between lamellas. And in the immediate space between lamellas, we have bacteria. This is how David and I were actually linking our work. Fish have a microbiome in their noses. And what you see here, we expose, in this case, goldfish to 0.1. It's a very, very, very small concentration that it's very, very, very common in our waters. And 1 millimolar, that it's a little bit higher, it's stressing them a little bit, but it's not making big, big damage in the fish. Every one of these columns, it's a single fish, and every one of these colors is a different genus of bacteria. You can see that the most part of the controls, they have this genus that is green, and I'm not gonna say all the names, but we'll have Pseudomonas, Yersinia, and all this. I'm not the microbiologist in this collaboration, but you can see that they have different groups that they can be repeated in every individual. What happened when we expose fish to 0.1? You can see a shift of the populations. You can see that some groups disappear, like this purple and gray. You cannot find it here in any individual. And you see new colors, like green and blue. So you have like Gemata and this one here. I don't want to say the name. So you see that there's a change in the microbiome. Microbiome is important for the physiology of any vertebrate. They are probably helping in this olfactory function. And if not, they are at least being like a barrier, a defense for fish, isn't it? So this is a big change in a very, very low concentration. The polluted waters around us, they are already affecting our fish. Also, I measure how nitrate accumulates at very, very, very small concentrations of nitrate, and then at some medium ones. And you can see that in the nose and brain, the accumulation of nitrite is like 2, 3 times, sometimes 10 times in the nose and in the brain. Only in these two. In the case of gill, liver, and kidney, these treatments are too small. And the tissues are doing okay. This is the these concentrations are the normal endogenous concentration of nitrite because it has other functions in these organs, but they are not changing due to treatment. So not only nitrite is affecting the nose, it's only affecting the nose and the nervous system in fish. And I can go further and just check again the histology for these very small treatments. Again, you can see here an olfactory epithelia. Every one of these little dots, these pinky dots, are actually olfactory neurons. You can see the brass border with the mucus, the electric blue mucus. And in this micrograph, you can see that the epithelium is kind of broken. You can see like holes. You can see also that the cilia is increasing. It's like 2, 3 times longer. They are they are not in danger. They're probably able to smell, but they're in an inflammatory process. So they're already sensing something is going on, and probably they are having all these processes to renew all these olfactory neurons are put there. Also, we check if this is affecting the tissue, the nose. Maybe it's also affecting the olfactory function, because we know even if it's a little bit irritated our noses, we can still taste coffee and chocolate, isn't it? So let's see if it really affects the function in fish, and if it's actually something that can damage our natural populations. So again, I did this, my students did this experiments in which they put, again, these goldfish here in this two choice maze. We pump water in one side and a vacuum in this other side. And we measure something that is called index of preference. Index of preference is positive when fish have preference for the cue you're interested. And if it's significant, higher than 0, you can see here the asterisk, that means that the fish is going to do the food cue. So obviously control fish are attracted by food. Fish at 0.01, they're actually more attracted for food. And I still don't know why is this happening. And 0.1, they're still attracted by food, but at 1, they lose. You can see there's not significantly different by 0. This is only in 2 weeks. At 3 or 4, actually, you can see that this effect in 0.01 is not there anymore, but 1 millimolar already lost the ability to detect food. 1 millimolar is something that you can find in nature. So this fish is not able to discern food. It's not gonna die instantly, but in the long term, they're gonna have problems to find food, and probably is affecting their survival. Also, I found a very, very interesting effect. It seems that nitrate at very, very low concentrations is actually helping the nervous system. And I'm not going to speak about that, but I'm telling you that I'm developing another project just trying to understand why nitrate they have this double-edged sword effect, in which very, very low concentrations is good, but increasing a little bit is bad. But we're going to center my talk only in bad things. I also found that nitrate, it's altering the neuron function. Neurons communicate by little molecules, can be in neurosteroids, can be peptides, and they can also transfer electricity. I was interested to see if neurosteroids released by neurons will be affected after nitrate treatment. Why neurosteroids? Neurosteroids are related with integration of movement and fish swim. So I thought, well, if it affects movement, that will be a big area of research. And they're also related with the activation of the immune system, which is also important to face this inflammatory process. So I found, we found in the lab that these steroids, neurosteroids, can be modulated by nitrate treatments. So in conclusion, nitrates can affect the sense of the smell in goldfish. And one might say, well, who cares about goldfish? Goldfish is this fish that we find everywhere that Aunt Peggy just throw in the bathroom, and now it's an invasive species in Texas, so it's okay if they die, isn't it? You have this huge monster goldfish in the river choose affecting our natural community. So I wanted to show an example on how basic research and physiological research can be translated to something that can be functional, something that can have output that is beneficial for humans. And one of them is aquaculture. So I transfer all this knowledge to aquaculture of catfish. Do you guys like catfish?
Yum, yum, yum.
Yum. Catfish is the most cultured species in US. We produce catfish in the order of millions, and it's also an industry that produces dollars in a bunch of jobs. But they have a problem, because we want to produce a bunch of protein in huge density systems. They feed, and they accumulate ammonia, transport nitrite, and they also have health problems related with bad quality of water. The main pathogen affecting catfish is this guy here, this bacteria called Capsula pylori, that induces this syndrome called enteric septicemia catfish. And you can see it's very, very nasty. The poor catfish here has a bunch of blood processes. They are just full of hemorrhages. They also infect the fish through the nose, creating these kind of holes in the brain. You can see the brain in this fish. They can also induce some kind of inflammatory process in the belly. They have this bell belly and very nasty ulcers in the tails. So I was thinking, I know the catfish have problems with this. Sometimes they can lose the full culture, the full farm can go down with this infection. Not only this, we know that the low water quality is related with more episodes of these infections. And specifically nitrite, it's related with this. So I was thinking, what about, and it's one of my students is working on that at this moment. What about if this healthy microbiome in the nose, it's protecting catfish from the invasion of this bacteria? Oh, you love that. But when we have nitrite, this microbiome is altered. And I knew that catfish have alterations in the microbiome. And then this bacteria has a portal of entry to the brain, and that's what they die. So let's go and try this. First, I wanted to see, well, not only one to find a problem, I also want to generate any kind of technology that can help to fight this disease. So if we have a problem with the microbiome, yogurt, probiotics. So we said, well, we're going to try to isolate bacteria from the nose and create probiotics. And then we're going to feed to the fish. So we have fish that are happily in clean water, fish that are fed with these probiotics that we design in our laboratory. We have fish with both probiotic and nitrates. And then we have fish that are only nitrate. And you can see that the concentration is pretty low, the one that you can find in a farm. And we put the fish for 60 days in these treatments. And after that, we will do what we call a challenge. We will expose catfish to this Edwardsella and see what happens. So here I'm gonna show you how the part of designing probiotic work. We culture this pathogen in our plates, and we also culture the different isolated populations from the nose and actually the gut, because we are full of this bacteria in the gut. And then we do a challenge in which we try to see if the growth of this pathogen is affected by any of these populations that we find in the nose and the stomach. And you can see that the catfish nose doesn't do much effect, like the Edwardsella pylori is still growing. So not a good candidate for probiotics, but there's a bunch of bacteria that it's affecting the growth of this pathogen. And also we still have bacteria microbiome from the goldfish, and it seems that they're very, very effective to fight this. So we actually, I only show like few of them, but we try this for hundreds of bacteria we found in the stomach mainly. In the nose, they don't have that many. So that was very, very difficult. And just tiny noses don't have that much bacteria. Well, the next thing we did is, let's see if the nitrate treatment is working. So this is my student that is doing her master's in my lab and studying all these things. She's also an artist, and she's the one that is designing all these figures you are seeing in BioRender. By the way, I recommend you BioRender. It's for free. They don't pay me, so but it's a very, very good tool to generate figures for your presentations. So I found that actually after a couple of months, we have a slight increase of nitrate. And let me tell you, this increase is not really important. It's not going to affect the physiology as much. And you can see a little bit more in gill, but after 30 days, they're going down. So fish look good in this treatment. And when I was looking at them, they're happy, they're eating. So even if they're exposed to nitrate, they look okay, isn't it? Not so much in the case of the nose. In the nose of a catfish, I love catfish noses. They're huge and full of lamellae. And I'm a nerd. You can see that maybe I'm the only one that likes noses. So you get all these pink dots are the olfactory receptor neurons. Catfish has a very hairy olfactory epithelia. All these little darker cells are pluripotent cells that are going to give new neurons. I don't know if you can see some lines here. These are actually the axons of the neurons, and some cells here in the middle will be red blood cells. What happened when we expose catfish to 0.2 millimolar for 60 days? You can see that the mucus layer that was very, very thin and conspicuous becomes thicker. The cilia becomes longer. The epithelia is the structure, and you can see these dark cells actually intruding in all parts of the epithelia. They're probably white blood cells or macrophages, just eating all these neurons that are dying. And the interspace is actually thicker. This is an inflammatory process. So catfish that actually they're very resilient and very resistant to nitrite, they're already being affected in their nose. The same for their stomach. I wanted to show you their stomach. This is a normal stomach of a catfish. You can see in less magnification, these are all the different folds of the stomach. The electric blue is in the production of mucus by the Krebs cells and a very, very low pH. And you can see in a higher magnification, these endothelial cells that are like columnar cells, the different folds, and the production of mucus. That's how a catfish healthy stomach looks. You see, you didn't know? Now you know. But when we expose this fish to 0.2, can you see the difference? It's dramatic. The folds are really not, they're kind of random. You have a bunch of production of mucus. You have a lot of cell death. You can see some holes. The Krebs cells are actually being bigger. This is a very, very small concentration of nitrite. Is this affecting also the physiology, their function? And in this case, what I wanted to do is measure the olfactory signals in the nose. So Avida was introducing me as electrophysiologist. I put electrodes in the nose and I measure these electrical signals that the neurons elicit after detecting chemicals like food, like amino acids, like alanine and glutamine that are in food, or bile acids like tarocolate or just fluids, bile fluids. So I put these electrodes here in the nose and I add whatever odorant, and I see just action potentials or receptor potentials. And I don't want to put all the results about this. I want to just to summarize. But I found something very interesting. It does change the olfactory function in fish. But what I was expecting is that when you put nitrates, everything goes down. And when you have probiotics, everything is okay. Well, it seems that the combination of nitrates and probiotics are as good as the controls. So they have very powerful signals, sometimes even more powerful than the controls. Again, this is probably some interaction of this good effect of nitrates and very low concentrations, small concentrations. So I'm still thinking about that. It seems at least that in the probiotic treatment, it's helping to smell catfish bile. And bile fluids are involved in this pheromone communication I was telling you about sex reproduction, and catfish probably communicate with their bile compounds. So that's good. It seems that the probiotic treatment are helping them to reproduce, not so much in terms of feeding. But they make also, the nitrate treatment make also more sensitive. What means sensitive? It means that they can detect these odorants at a smaller concentration. And for some reason, the nitrate treatments increase the sensitivity. And I'm still thinking about that. I'm not open to suggestions. Something is going on. There's some effects of the probiotics, but we still don't know what's going on. What I can tell is it changed the function. And it kind of still smell. Not only they kind of still smell, sometimes they smell better. But what happened with in terms of protection towards pathogens? So if they smell better, they are less protected or more protected. And these are other preliminary data that Ashley produced like not a long time ago. We exposed the fish to the different to the pathogen, and then we expose them to a concentration of pathogen that doesn't kill all of them. Because if you have a huge concentration, it's going to affect every organ. You don't know exactly if the pathogen comes from the stomach or comes from the muscles. So we wanted something that affects them. So they produce some ulcers and some symptoms, but they don't. They are not totally blown out by virus. And what we can see is if we measure how many individuals are affected by these pathogens, so we can culture all of these noses, 100% of the noses and 100% of the brain are infected by this pathogen in a control treatment. But this is the cool thing. If they have probiotics, they're actually much more protected. The nose is only infected in 30% of the individuals. And we didn't find anything in the brain. Success. Awesome. I'm going to tell you something negative about this later. What happened with nitrite? Actually, it protects a little bit. You can see that with nitrite, we have also a 100% infection and a little bit less in the brain. But when you have probiotics, this infection also diminishes. So that means that in environments that we cannot avoid nitrite because PSP and they are cultured at high intensity, and we cannot just flow water like crazy. We cannot put nitrates at 0. We can give a cheap treatment that we can add in the food, and we can avoid infection that can kill your full farm. The only bad thing that I can tell you about this is that the fish hate food with probiotics. It seems that it tastes terrible. Most part of these bacteria are actually aromonas that are sometimes pathogens too. So we only try like 10 different populations, and we are trying to find more money to design better probiotics. But we know they work, and if we find a probiotic that they love, maybe we have to put some shrimp and chocolate there. And it's something that attracts them, and that will be a fantastic treatment. So in conclusion, all this basic research allow us to identify that nitrates at low concentrations affect the sense of smell, which is very, very important to find food and produce and everything. But not only this, because the nose is the portal to the brain. Probiotic treatments that increase the health of this nasal mucosa are going to help to pathogen infection. And you can see here these pretty fellas. We have a catfish farm here in San Marcos and are very, very happy to see that we can produce this. They are actually, the farmers are waiting for us to produce the right probiotic for their fish. So with this one, I'm gonna just finish my presentation. This is my lab. I have a couple of my students, actually Whitney, she's the student that did all the goldfish work, and she's now doing her PhD in San Antonio, in UTSA. And the rest of my students, you can see that it's a very diverse lab. I'm promoting women in science and Hispanic in science. And actually, well, these two PhD students are from Bangladesh, so they just put down my statistics, but they're still a minority. They're my international people. And we have a fantastic lab with master PhDs, undergrads. We have a lot of fun. And you are welcome to follow my activities. I'm kind of active in Twitter, not in other social media. And if any of you is interested in continuing doing a master or PhD, please let me know. I hope I convince you how important is to understand the sense of the smell, not only to understand ourselves, but also to detect and have a tool to do management of natural species. I also want to say thank you because all this is well-funded by governmental agencies. And with that, I will open for questions.
Just curious, of the histological figures of the rosette, it's occluded and whatnot. What does this look like developmentally? So if you just leave them chronically in this, as the fish grow, does the organ become deformed, or does it modify? or what does it do?
That's a very good question. More than 2 months, I don't know what's gonna happen. I can tell you that this have an amazing potential to just change the whole nose and recover in the terms of days. So probably if they are not affected by nitrates anymore, they will recover and that's it, totally. So you can remove the full rosette and they will grow on time. But if they have a continuous exposure, it can be two things. They just chronically lost the sense of the smell, or they are who knows? Because we're exposing fish for years, all this. So it can be that they adapt their nervous system. Probably they are more resilient to the concentrations of nitrates we have in our aquatic environments. But more than 2 months also to expose fish is torture for my students. You know, they have nitrates every day. So 2 months is good enough for a chronic experiment, but who knows? I'm very, very interested to see what's happening in natural populations in which I know they have a high concentration of infection. Let's see. Yeah.
I just thank you for this. This was fantastic. I am. Thank you for expanding to that. I work. One of the projects I work in is with patients, human patients that have a multiple chemical sensitivity disorder or TILT, which is toxicant induced loss of tolerance. And basically, a lot of times, the story with many of these patients is that they've been exposed to some, often pesticides is a big one, and either an acute event. And then what happens is, and we think it has something to do with the sensitization of mast cells. And then they get sort of, then they become hypersensitive. So what happens with these, then people develop aversions to certain specific smells that they didn't have before. Yeah. So when you mentioned that odd hypersensitivity to smell, we're seeing that in human patients who have this syndrome. And unfortunately, because the pathophysiology mechanism is not well known or understood yet, many of these patients get bounced around from doctor to doctor and they end up getting sent to psychologists because, you're imagining.
So do you think it's at the level of the nose or the level of the brain?
I think it probably affects both, because I think there's there's a variety of, we have some tools to measure symptomatology that have been validated, and respiratory. There are cognitive, like brain fog effects. A lot of times is very common. The olfactory, it's affecting a lot of different systems, and I do think that there is an overlap with some affective disorders. So, which again, so teasing all that apart is tough. But I just, it's got me thinking. Just want to thank you.
I have a hypothesis about how it is happening. I think we have an effect in the level of the nose. You remember the cilia? Okay, this is an inflammatory response.
Yeah.
But you have the olfactory receptors in the cilia, so you have more surface for the receptors to find chemicals. So I think that's why they become more sensitive and they can detect more others. But there's also what happens in the brain. We learn a lot with COVID. So COVID can rewire brain in a way that some people that is recovering from anosmia and COVID, they can smell coffee and they think it smells like food, literally. It's food, and they have a rewiring with the food sense. So I think inflammatory process can affect your nose and can affect your brain, and it's fascinating. We're learning so much in the last few years.
Well, it's just, I mean, then the idea that there are potential, because that's another part of this is looking at firefighters exposed to smoke and looking at the hopefully funding with the nasal microbiomes changes. But the idea that there could be food or spray or something like that could help these patients is massive. I mean,
Yeah, and I'm hoping that if any of you go to biomed and with humans,
Yeah.
I think the research in microbiome in the nose is something with huge potential. I think I cannot stress enough how important is the sense of smell in humans. Actually, problems in the sense of smell are related with dementia. And even I think, medical doctors say if the patient, all patients are losing the sense of the smell, it's kind of related with the time of death. So it's correlated with fail in your neurobiological function. So, yeah.
The NIH funding in the future.
Yeah.
Yeah.
New model.
Yeah, I'm trying to sell my molecule. Yeah, so I send a proposal. But actually, now I'm not focusing as much in what happened in the brain, and I'm going for pregnancy because I noticed that we have nitrates in water, and this can be accumulated in pregnant women here in Texas. And how is this affecting pregnancy and the development of the nervous system? So I have a model in my lab. It's a fish that is live bearing, so I'm getting pregnant. And they give birth to little fish. And we're actually trying to see if the nitrates affect
Yeah.
the nervous development. So if it's funded, I will tell you.
That's
here.
good. Yeah,
Let's do.
there's some suggestion that it's low birth rate.
Yeah.
Low birth rate.
So to be pregnant in Texas.
Yeah.
It's happening.
All right. I'm sorry, I got to kind of, because there's a class in here in a couple of minutes time. So if you have any more questions for Dr. Huertas, please feel free to grab her afterwards. She'll be going for lunch with the grad students. We could have whatever you want, as long as they make it down there at least. And yeah, thanks very much.
Thank you.
Thank you for having me. You were right in the shop.